So one thing I think is really interesting is in thinking about Vibrio as basically a vessel for all of these phage-phage interactions and competitions, right? And so I think that the element that we study called PLE, which the phage uses the CRISPR-Cas system to overcome, can almost be thought of as a phage, right? It's embedded in the genome and it responds to phage infection, and it actually steals that phage's packaging machinery to spread itself to neighboring cells. So it's a parasite of a parasite. Right? And I think that Vibrio is really the vessel there, right? And the really interesting, cool molecular, amazing molecular piracy that's going on is happening all between these phages. And I think it's also very interesting to think about how these filamentous phages that are directly involved in virulence in Vibrio are responding to this battle that's going on in the same cell that they're occupying. And so we haven't seen any evidence that they are responding, but because ICP1 has been around for so long, I think that it wouldn't surprise me if we found out that indeed it is responding to this threat to its vessel, which it's trying to preserve as well. So essentially cholera is driven by a phage, right? And so it's a filamentous phage that is integrated into the genome and basically donates a virulence factor to Vibrio cholera so that Vibrio can cause disease in humans. And that ultimately results in increased fitness for the virus, because the virus is a part of that bacterial chromosome, and then that toxin ends up allowing Vibrio to replicate and spread in that niche. So it's all about phage when it comes to cholera.